In this video, I'm going to explain the standard level genetics, monohybrid inheritance, codominance, multiple alleles, and sex linkage. Firstly, a reminder of chromosome structure. Each chromosome is made from two chromatids, which are genetically completely identical. In their pairs, we say that the chromosomes are homologous. This means that they carry the same genes, but not necessarily the same alleles. For example, you could have tongue rolling here and here. This chromosome could carry the can allele. This chromosome could carry the can't allele. This means that for every gene, you have two copies or alleles, one from each parent. Within those pairs of alleles, the alleles can either be dominant or recessive. If you have a dominant and dominant allele, then you will have the dominant phenotype. If you have the dominant and recessive allele, you will have a dominant phenotype. If you have a recessive and recessive allele, then you will have the recessive phenotype. These two characteristics are what we call homozygous, because they are both the same genes, whereas this one is heterozygous because you have one of each. These represent the genotype, so which alleles you have, whereas these represent the phenotype, the characteristic that comes out. If we cross two yellow peas, what will the offspring be? Well, in this cross, we will say that yellow is dominant over green. When we define the alleles, we will say that this big Y represents yellow and this little y represents green. You always use the same letter to represent the gene, with the capital being dominant and the lowercase being recessive. Do be careful when choosing your letter to make sure that the capital and the lowercase do look different. So we have two yellow peas. We're going to have the heterozygous one. So our phenotype, our parental phenotype, is yellow crossing with yellow. And because they are the heterozygotes, then it is big Y, little y crossing with big Y, little y. Now when we come to make the gametes, our gametes are obviously haploid cells rather than diploid cells, so they can only have one chromosome in each one. So we will have a big Y gamete. You always put the gametes in circles. A little y gamete from this parent, and then a big Y gamete and a little y gamete from this parent. So to work out the F1 genotypes, we do a Punnett square, where we put the big Y, little y, big Y, little y. So this is going to be big Y, this is going to be big Y, little y, this is going to be big Y, little y, and this is going to be little y, little y. So our F1 phenotypic ratio is going to be 3 yellow to 1 green, which is the standard Mendelian ratio for a monohybrid, one gene, heterozygous cross. Codominance occurs when two alleles are expressed equally in the phenotype, giving a blended characteristic. This is notated slightly differently, with the capital letter being used for the gene and then a subscript for the alleles. You often find this in hair colour of animals, so we might use the capital C for the gene coat colour, and then if we had, for example, CW for white and CR for red, what you would find is that they are both dominant, but where you have the heterozygote, CWCR, you get what we call roan, which is a codominant characteristic. There are times when there are more than two alleles for a gene, and blood group is a good example of this. We notate this with a capital I for immunoglobulin, and then superscript letters for the alleles. So we have I big A, we have I big B, and then for the third allele, you either write it as IO or as little I, and this is the recessive allele. So the A and the B are both dominant, in fact they're co-dominant, and then the little I is recessive. So we have 4 blood groups, A, B, AB and O, and A can either be IA, IA, or IA little I, B exactly the same, IB, IB, or IB little I, then AB is IA, IB, and O is the recessive, so little I, little I. This means that if you had these 2 parents, you could end up with each of the blood groups in the F1 phenotype. Sex linkage occurs where a gene is found on one of the sex chromosomes. In reality, this is usually the X chromosome, but it could technically be the Y. Haemophilia is a good example of this, and it's notated like this. We use the X to represent, or the Y to represent the chromosome, and then the superscript letter to represent the alleles, the capital being dominant and the lowercase being recessive. Colour blindness is notated in exactly the same way, but with a B instead of an H. Note that the Y chromosome does not carry an allele because the gene is situated on the X chromosome. Consequently, these disorders are far more likely in males who only have one copy of the X chromosome, and therefore when they get the recessive allele, it is always expressed. Remember, boys always inherit their X chromosome from their mother, so when looking at the pattern of inheritance, you can spot that it will be going from mother to son. Pedigree analysis allows us to follow traits through generations. A circle represents a female, with the filled in representing having the disorder, and a square represents the male, likewise the filled in represents having this disorder. This setup represents an autosomal recessive disorder. Both parents are normal, but they have a sufferer child. If you went up the generation here, you would probably find that these had perhaps a sufferer parent, or there is a sufferer somewhere in the generations. This one represents potentially a sex linkage situation, so you've got normal parents but only the sons have the disorder. In this one, this woman would have a sufferer father, and that would be where she had got her recessive X chromosome from. This setup represents an autosomal dominant disorder where a parent has the disorder and so does at least one child. This is because this person would pass on at least one dominant allele, so some, or if they were homozygous, all of their offspring will have the disorder.